come in, please come. Please be seated. Hello.
Hello, ma'am.
How are you?
We are fine, ma'am.
Okay. Tell me, what can I do for you?
We are here to know more about clinical SAS programming.
Okay. And may I know your basic qualifications?
I have done MTech in biotechnology.
Okay. And how about you?
I have completed MSc in microbiology.
Okay. So what made you think about this career?
Ma'am, because traditional careers are available after graduation in microbiology or pharmacy, like jobs in QA, QC and R&D, but we get paid very less, and at the same time jobs are not available in the nearby cities.
Okay. And how about you?
We want to enter an IT company, but at the same time we don't want to go away from our own field.
Okay. So first, I will explain you what is clinical research. When you will know about the flow of clinical research, you will be easily able to understand different careers in clinical research. So you must be aware that before the drug comes into the market, you have to make sure that it is safe and effective. Unless you make sure that the drug is safe, you really can't introduce it in the market. So do we directly use drug on human being? No. First, you have to use it on animals. You have to use it on animals and make sure that the drug is safe and it is causing the effect that it is intended for. So this is called preclinical trial or preclinical research. So once the efficacy and the safety of the drug is proven on animals, then only you can use it on human beings. And when first time you use it on human beings, it is called clinical trials. Clinical trials are done in phase-wise manner. There are four phases of clinical research, and with every passing phase, you consolidate on your hypothesis. Okay? So the first phase of clinical trial, you have to use very little number of volunteers, just about 10 to 30 volunteers. And in phase 1 of clinical research, you study pharmacokinetics of the drugs. When the drug passes that phase, that means the effects of the drug in phase 1 are proven, then the drug enters into phase 2 of the clinical research. In phase 2, you have to include little more number of volunteers, say about 80 to 100 volunteers. And this is called therapeutic exploratory trial, in which first time you will include patients or the volunteers suffering from that particular disease. For example, if your drug is antihypertensive, you will include drugs suffering from hypertension, that is high blood pressure. After the drug passes phase 2, drug enters into the largest phase, that is called phase 3 of the clinical trial. This is a large phase that is multicentric and it is global, which means at a time it is conducted in many hospitals. And the number of patients that are included in phase 3 are large number. That is, you have to include something like 5,000 to 6,000 patients in phase 3 trials. So basically, why are you conducting trials? You want to collect data, lot of data. And most of the times in phase 3 trials, the trial design is such that you have to divide patients in 2 arms, and one arm is called control and other arm you are giving the drug that you want to prove its efficacy. Okay, so every time when the patient comes for the scheduled visit, you carry out lot of investigations like vital signs, like some lab reports. If it is the oncology patient, that is anti-cancer drug you are studying, then there will be lot of other reports like MRI, CT scan, so many things. So every time when the patient comes, there are lot of data points that are created. The data is so huge that you can't depend on Excel sheets. Excel sheet will have its own limitation. So you must have a sophisticated tool to manage this data. And there are a lot of tools available like Oracle Clinical, Rave, Inform. So this data has to be managed properly because the data has to be clean. There cannot be any discrepancies in data. Now you will ask me, what is the importance of you being life science graduates to manage this data? You will understand the importance of data. For example, if you are working on a trial that say to increase hemoglobin, and if there is a value that says hemoglobin of 130, then you being a life science graduate, you will understand that there is something wrong with this data. So you will say It is not 130, it is 13.0. So one decimal point is missing. This is a very simple example, but because you are a life science graduate, you will understand that this data is correct or incorrect. Okay, so coming back to data management, you require sophisticated tools to work on data management, and there are good careers in data management also, and you get jobs in ITES companies. ITES is Information Technology Enabled Services. And the next step is, after the data is collected, database is clean and it is locked, SAS programmers come into picture. Now in data management you collect plenty of data, but at a glance will you be able to find out what does the data say? No. For that you require special tools. You use SAS programming and you work on that data, and the huge data is converted into tables, listings and graphs. So at a glance you will be able to find out what the data is trying to tell you. Okay, this is very important tool. And then you will ask me, is there any other tool available? There are some tools available, but US FDA has made it a mandate that if you have to submit the clinical data, you have to use SAS. That is the reason why there is so much importance for SAS programming. And after learning SAS programming, you will get jobs in IT companies, in pharmaceutical companies like big pharmaceutical sponsors like Novartis, GSK, Abbott, etc. You can also get jobs in big CROs. Do you know CROs? Contract research organizations like Quintiles, PPD, iGet. So after doing SAS programming, you can launch your career in any of these companies. So in short, after learning clinical research and all its allied operations, you can either work in sites, that is in hospitals, that is clinical operations. You can choose to work in clinical data management, or you can choose to work in SAS programming. There is one more career that you can pursue after clinical research training, that is in pharmacovigilance, that is tracking safety of drug all the time. So this is about the careers. Do you have any questions?
Yes, ma'am, but we do not know any programming language. We are not computer experts.
That's okay. Do you have smartphone?
Yes, ma'am.
Okay. So when you got your smartphone for the first time, was it easy to use?
Yes, ma'am. It's very easy and interesting also.
Okay. So it is just the same. It is as easy as using your smartphone. Of course, you must have interest for it. And one more thing I would like to specifically tell you, that it is not as difficult as programming in C++ or Java. It is absolutely simple. Of course, you should not have aversion for computers. What I'll do is, I will request our trainer to show you how SAS programming is done. Do you want to see that?
Yes, ma'am.
Okay, so come, I'll introduce you to our trainer. Kaveri, these are two inquiries. She's Sneha and she's Anjana.
Hi.
They are not from IT background, so they are little apprehensive about the way SAS programming is done. So will you please help them?
Yeah, sure.
She's our trainer. She's Dr. Kaveri. So she'll explain you everything. Okay? Okay.
So you are heading towards future in programming, SAS programming.
Yes.
Yeah. So all the best to you. And if you are feeling that whether I will be able to do it or not, so see me. I am a doctor, and still I was extremely interested in SAS. I tried it and I found that it is extremely easy to write the SAS syntax, and it is very close to the way we think. So you'll find it easier too. So I'll show you. Okay.
In clinical research, we get enormous amount of data, and SAS programmer has to generate tables, listings and graphs from that data. So to generate those, we have two building blocks in our SAS programming. First is the data step and second is the proc step, that is procedure step. Now here I am submitting one data step in which I am defining my column headings as UID, site ID, age, gender, height and weight of the patient. After that, based on the values of age of the patient, I am creating another column which is age group and I am conditionally assigning the values of age group as young, middle, old based on the values of age. Later I am also calculating BMI of the patient, and after that resides is my data. Now I am submitting this data step. I'll have to select the syntax and then click on this submit icon. Now what you see is in the Explorer window, here is a data set getting generated. Data set is nothing but a table in SAS. If you double click that data set info, you will see that the table is opened in the view table window, and you will see the column headings as UID, site, age, gender, height, weight, age group is also there, and BMI is also there. Now next I am submitting one proc step, that is procedure step. Here it is proc sort, which sorts the data. So I want to sort the data as per age, ascending order of age. So let us see. Here I am selecting the step and again submitting it. Now you can find that the new data set info_std is generated, and you will get message in the log that there were 10 observations read from the dataset work.info, and the dataset work.info_std has 10 observations and 8 variables. Now if you open that dataset, which is info_std, the sorted one, you will find that it is storing the observations as per the ascending order of age. We can also generate pie chart using SAS. Here I am creating one pie chart and I want to see the age-wise distribution of my population. And here is my output. So I can see that 4 patients are from middle age group, 4 from young and 2 from old. I'll get this output just in one click. Here I am plotting BMI age group wise, and I want to see how many patients are above the reference line of 30. And I get this output with one click, and I can see that there are 2 patients from young age group which are above the reference line of 30. Now I am having the data of drug sales, drug ABC and PQR across 3 different sites, Pune, Mumbai, Bangalore, and the data is quarter wise sales. So quarter 1 and quarter 2. And here is my output, which I will get after 2 steps, and I can see that bars are generated showing the drug sales across 3 different sites.
So just now we saw how easy it is to program. I hope that you must have got some idea.
Ma'am, we are really impressed. It was easier than we thought. Thank you.
Yes, ma'am. Thank you.
Thank you. I'm very sure that you'll enjoy learning too.